Hello, welcome to our next part of Unit 1, Biology, Grade 11. I think this section, this part is the last part. Talks about how technology is helping the science of biology in many different fields, and it gives you some examples of the technological devices. Of course, there are lots of technological devices that help the science of biology in different areas for research purpose, for their use in the medical field. Medical field, of course, those materials or those technological innovations or technological machineries, whatever that are listed in your textbook are concerned with those used in the medical field, medical tools. Most of them are medical tools described in your text. Otherwise, there are research tools used in the science of biology for research purpose, scientific investigation purpose. There are laboratory tools as well as field tools that are used for research in the field and research in the laboratory. Therefore, the others are those lists of instruments or machineries or materials, devices used in the medical field. And we can start this section by stating this technology provides us with tools used in biological sciences. What are these biological? This huge biological sciences refers to huge area of investigation, study, or application. Biological sciences, applied biological sciences, as well as uh, pure biological sciences, which are focused on research. Therefore, both use tools uh, made for their purpose, their research or the application of the tools in order to diagnose or treat uh, patients in hospitals. When it comes to these tools, the first list, medical tools. Only these are medical tools. Most of them are medical tools that are listed in your text. The first tool is this tool. You can see this tool. This tool. This tool is known as commonly used digital thermometer. There are many different brands, different types made by different factories around the world. Therefore, digital thermometer used to measure body temperature in hospitals. Of course, digitalizer just without contact with your body, it can measure your body temperature, core body temperature, the inner body temperature, and it's therefore this is digital thermometer, one instrument used in biological, medical science, medical tool as medical tool used as medical. The other is pregnancy urine test. Pregnancy urine test, similar. It's kit that is composed of chemistry, some sort of chemistry. Antibodies are there. Antibodies react with this hormone, in order to test. This hormone, by the way, this hormone is human chorionic gonadotrophin. This hormone is produced by the fetal cells or the if there is pregnancy, the baby becoming cells of the zygote in the placenta, in the uterus of the female. If she is pregnant, this hormone is produced within 2 weeks of pregnancy. 2 weeks of pregnancy, human chorionic gonadotropin. Therefore, if a female needs to test whether there is pregnancy or not within these 2 weeks, this is tested. This is secreted by the embryo and it's found in the blood. It's found also excreted in the urine. You can take blood or urine. Since urine is cheap and it's a waste, of course, metabolic waste, take urine. That's why it's known as usually pregnancy urine test. Otherwise, you can use blood also because the hormone is found. The aim of this test is the chemical reaction is made between human chorionic gonadotropin hormone and antibodies which are found in the kit. The kit is this one. The kit is this one. Then they react and produce color. This one is positive change. Positive, negative. Uh, this one is positive pregnancy. Positive pregnancy, this one is negative. Positive means the hormone is found in the urine. That means she is pregnant. That means, uh, therefore, the baby is growing inside her uterus. Then the next decision will be, it's up to her, of course. Therefore, the test result in one line is control. Of course, this one is control. That means these are antibody cell. If you do get color change, you put the urine here. The urine sample is put here, and within 3 minutes of time, it will show you this double color. If it is positive, that means if the hormone is found in the urine of that female. Otherwise, it remains like this. If you put urine here, if the urine do not contain this hormone, that is HCG, and therefore there is no antibody. There is no antibody antigen. HCG. There is no HCG. There is no HCG means there is no reaction. If there is no reaction, you will not see this strip here, the color change. Therefore, diagnostic purpose. This kit is used for diagnostic purpose. The similar instrument which works based on chemical. Such instruments are known as biosensors. Biosensors. They sense. They give you a result based on biological molecules. For example, in case of this, what are reacting here? Antibodies specific to HCG. Antibodies and HCG interact to produce color change. Therefore, it is biological. We call it biosensor because although this is mechanical, this is plastic or any metal, whatever it is, this is physical component. But inside here there are biological components. What are the biological components of such instruments? Of course, antibodies are found there. Of course, the urine is biological. It contains biological materials like urea, other waste materials, water. It may contain other chemicals. If she is pregnant, the urine of pregnant female also contains HCG. So if HCG is found, it can sense it. There is chemical reaction that gives you color change. You observe it and you say it is positive. If there is no color change, negative. Negative result means there is no in medical science. Therefore, positive test means there is. Therefore, the other biosensor, biological sensor is this instrument. This is known as glucometer. They call it glucometer. It measures the amount of glucose in your blood. Just from it takes glucose. It checks the glucose in a small drop of blood. As you see, you pierce it. When blood comes, a drop of blood comes, you touch it. Glucose diffuses, and this calculates the amount of blood, not in this drop of blood, in your body. Imagine, totally in your body. What is the amount of blood per deciliter of blood? That is 90 to 120, the normal range. Therefore, if when you test it, this is for diabetic mellitus, DM patients, when you test it by piercing here, your blood, when you test it, if you find your blood glucose level is, say, 200, oh, it's a big deal, 200 milligram per deciliter, oh, this is abnormal. The normal range is 90 to 120. Therefore, you can check it. Therefore, if your glucose amount of glucose in your blood is very large or going up, that is hyperglycemia, you have to take your own measures. You may take your injection of insulin or any other drug, diabetic mellitus drug, which can reduce your blood glucose level. If it is below, you have to take some sugar or some carbohydrate foods in order to bring it up. Those are not good. That means hypoglycemia, that means lower than normal, and hyperglycemia, larger than the normal range, the highest range. Therefore, This is very important. Every diabetic mellitus patient should have this glucometer, a biological sensor. It checks every morning, every evening, checks the blood glucose level so that diabetes mellitus will not cause the distraction it causes. Oh, it's very, very, very uh, dirty, I call it, dirty disease. It's diabetes mellitus damages your nerves, it damages your eyes, it damages blood vessels, all complication. It may damage your kidneys also. It's not good. Therefore, you have to control your blood glucose level. If you are diabetic, you have to buy this. It's very cheap, of course, little bit cheap, and so that you check every morning and every evening, midday. You can check anytime your blood glucose level, so that you can control the amount of blood in your blood and the amount of glucose in your blood. When it comes to the other is HIV kit. If your blood contains the virus, it can give you similar to the pregnancy test, the urine pregnancy test. And this one is the positive test, and this one is the negative test. And similar, two strips, positive, negative test. Therefore, positive means you have HIV, you are HIV positive, you have HIV in your blood, and this measures, similarly, it measures uh, the amount of antibodies in your blood. When HIV enters into your blood, your white blood cells produce antibodies. You see, the HIV is antigen, and your white blood cells produce antibodies. Therefore, antigen and antibody interact here to give um, color change, so that you can see if there is color change, positive. If no, negative. The other is microscope. As usual, this is a very, very essential tool of the biologist, or specifically the microbiologist. Microscope, of course, it is also used in the other fields of natural science, like physics and chemistry also. But most important tool of biology, especially cell biology, tissue science, histology, microbiology, these instruments are really smart, super smart molecules. And therefore, microscope, this is compound light microscope. This microscope is compound light microscope, and this is also found in high schools, the simplest form for the simpler form. There are also many different varieties of compound light. It works by light. Vision is formed by light, and used in diagnosis, hospitals, in research laboratories. Therefore, as teaching aid in schools, used compound. The other is this is where while students are observing through through compound light microscope, and the other is electron microscope, much sophisticated with the highest. By the way, the compound light microscopes gives you magnification power of maximum, the highest, the most complex type of compound light microscopes gives magnification power of 2000. That means it increases the size of a specimen that you are observing 2000 times. It has two lens systems. You see this objective lens system and also ocular lens system. Therefore, things are magnified two times. First by this, then by this. Anyway, the maximum magnification, this time this magnification is 2000, the maximum. Therefore, this is while using then electron microscope. Electron microscope, you can see there is one other property of microscopes, which is known as resolution. Resolution means the maximum distance, the two specimen, two microscopic things are close together, so that your microscope can show us two separate. Okay, I mean the minimum distance. The minimum distance. If two things, for example, maximum resolution of transmission electron microscope, two types of electron microscope, transmission and scanning electron microscope. The transmission electron microscope maximum resolution is 61 picometer. Which means this microscope can show you two things as separate two things only if they are with such distance, at least this distance is separated. If this distance is increasing, this microscope will not show you if this distance, if this distance is lower, decreased, or if the resolution power is lower than this, it will not show you two things as two. It merges them. So what do you call this? This is the minimum distance by which two objects or two specimens separated, so that your microscope can show these two things as two separate things. The meaning. This is minimum. Therefore, below this means you will not observe the two things as two separate things. You see rather one merging over the other. This is simply magnification. How much the size has been increased? Imagine it's very huge, 250 times magnifying. This one is. Then when it comes to the other type of microscope, the scanning, lower magnification, plus also lower resolution power. Resolution power one nanometer. That means this can show you two things as two separate things, only if they are separated by the small by this distance. Only if they are separated, so at least separated by this distance. Otherwise, if they are separated below this distance, they are seen as one, or one merging over the other. The magnification power as well as the resolution power is smaller scanning electron. Be careful. Resolution, don't worry. Be careful. The resolution power, the smaller the resolution power, the figure you see, the smaller the resolution power, the higher it is resolving property. Okay, otherwise this one is directly proportional. The higher the magnification number, the higher the ability to increase its size or magnify the object. Therefore, you can see here this scanner type. Anyway, these instruments are medical as well as research. Either you may carry the micro. Of course, these you don't carry electron microscopes to the field. Lab tools, they are lab tools, but you can carry your own compound light microscopes to the field. It could be field tool. Otherwise, you can have portable compound light microscope that can be easily carried into field for research purpose. When it comes to the other tool, computer information technology scanning, CITS, is a device used for investigating information on disease or cancerous areas of the human body, analyzing an image and processing it in some way. It analyzes image in your body, the body of patients, and processing it in some way, density, size, or mass, something like that, so that as it cancerous, those some ball of mass of tissue in your body may be assessed. And for this is an instrument that is computer information technology scanning. This scanner, it scans information about your body, the image, analyze it, digitizing real-world objects, document or picture, and converting them to digital information for a computer to store or manage. This is another purpose. You see, it can digitalize the information, document, medical or picture or written document into computer information, digital information for a computer to store or manipulate or process it, so that it gives you the result. Sending information to the computer and can't receive information from the computer like a printer. It's an output device. You see, sending information to the computer, and of course can't receive information from the computer like a printer. It's not just like a printer. It sends its output device. It gives. And the other instrument is computed tomography scan. Computed tomography scan. I will give you links, video links, how this machinery to enable you see videos how these medical tools are working. Computer tomography scan, CT scan, you know it, CT scan. This is a very important machinery or tool in medical hospitals or health facilities. Therefore, it is a sophisticated X-ray technology used to take many X-ray pictures of the body and used to detect and screen for a variety of disease and conditions. You go inside here, many X-rays from many directions. X-rays are released from many directions into your body, particular body, like if you have problem in your lung, in your lungs. If you have problem in your kidneys. Therefore, it takes pictures and analyzes so that the specialist observes the picture from the scanner, the scanning of this computer tomography scan, and tells you whether it is the specialist, the computer, the CT scan specialist, or the radiologist. We call them. There are specialist doctors specialized in this operating machineries and reading the picture taken from these machines. Therefore, therefore, positron emission tomography, the other medical tool, PET scan, CT scan releases X-rays into your body and takes image from the X-rays. This one, positron emission tomography, PET scan, is an imaging technology device used to check for diseases or information in areas with cancer, especially designed for cancer diagnosis. It uses a special type containing radioactive. You see. Now, first the patient is given radioactive tracers. You see, for example, the usual radioactive tracer is glucose. Glucose. The positron emission tomography, especially PET scan specialist, through injection through your blood, gives you a glucose labeled with radioactive element, like usually fluorine, radioactive fluorine. Glucose at one part contains what? Radioactive fluorine. So it is injected. Therefore, that radioactive, for example, if your body has cancer cells in your brain, if there are cancer cells in your brain, this radio labeled tracer, for example, radio labeled tracer is in this case glucose to which radioactive element has been attached, like fluorine, radioactive fluorine. It goes into your body, it's glucose. The only difference from glucose is you take one hydrogen from one carbon and add one fluorine to it, radioactive fluorine. Those areas of your body that contains cancer cells are voracious, or they are with huge appetite for glucose. Compared to the other normal cells, cancer cells use huge amount of glucose energy. They use high amount of energy, so that for that to produce high amount of energy, they have to take from the blood, they have to take large amount of glucose. So when such type of glucose comes in your brain through blood, not the normal nerve cells that take too much. Those that take too much glucose are the cancer cells. So when this radio labeled glucose enters to the cancer cells, this radioactive fluorine decays. It's decayed. When it decays, it emits radiation. Scientists are taking this into image. They are following. They are just this machine checks from which area of the body the radioactive activity is highest. You see, the highest radioactive activity, radiation emitting region is the region where there are cancer cells. So further investigation will be done. You may take sample of cells from that cancerous area and investigate additionally. In order to be totally sure, you further investigate whether these cells are cancerous cells or not. There are many other chemical tests. Therefore, not only by you don't just use positron emission tomography and say enough, it is cancer. No, You have to ensure it by other tests. It may tell you the region where the cancer cells are. They may be not cancer cells. Most of the time they are cancer cells because they are using huge amount of glucose. This is one property of cancer cells. They are voracious. They eat too much glucose because they use too much energy. So when you give such glucose, radioactive element containing glucose, these are the cells that take the highest amount of glucose that has been injected into your blood. So when this glucose goes to that area in large amount of radiation is emitted. That radiation is captured by your machine. Oh, it is in his head. Oh, it is in the lung. Radiation is emitted large amount. Oh, it is in kidney cancer if it is kidney cancer. The kidney, there are special areas of the kidney where there is high amount of radiation emitted from the consumed glucose. You see, this is positron emission. You see the difference? Positron emission tomography. The radiation is not emitted from the machine. When it comes to this CT scan, the machine is emitting radiation, X-rays into your body to take image. When it comes to positron emission, the substance injected into your blood is emitting radiation because it has been labeled by radioactive substance like fluorine, the usual code. Glucose has been labeled by radioactive fluorine, and this is the difference, you see. When it comes, it visualizes and measures changes in metabolic process. Metabolic process, glucose consumption, metabolic. Oh, these cells are high metabolic. There is huge change. They are taking large amount of glucose. So this area is really metabolically active. What does it mean? Metabolically active means if a tissue or a group of cells are metabolically active, they are consuming large amount of glucose from the blood. They are taking large amount of glucose to the blood. And you can easily know how, because the glucose has been labeled to be radioactive. Such glucose, when enters into such cancerous cells, from there is high amount of energy emitted, radiation emitted. That radiation is captured by image, and it can tell, the specialist can tell you it is from your head or your kidney or your lung or your liver, whatever region of the body where the cancer cells are suspected. The other is handheld body fat calculator. This instrument can calculate. You hold it like this. It calculates the amount of fat in your body, fat amount. Especially if obesity is a problem. This is good instrument to tell you, just like glucometer. It can tell you your amount of fat in your body. Fat is not good. It is a risk factor for cardiovascular problems, problem of the heart, problem of blood pressure, whatever it is. Therefore, handheld body fat calculator as a device, very important. The other is MRI, magnetic resonance imaging. This is a very gigantic magnet, very huge magnet. This magnet, similar with the CT scan, it emits magnetic. It is made of magnets, and it works by the same principle. Magnetic resonance imaging is a medical imaging technique mostly used in radiology and nuclear medicine in order to investigate the anatomy and physiology of the body, to detect pathologies including tumors, inflammation, neurological conditions, nerve problems, such as stroke disorders. If there is stroke, breaking of blood vessel here and clotting blood here, it can tell you by taking image. Blood vessels, among other things. Contrast agents may be injected intravenously or into a joint. And in this case, this emits radiation, just like CT scan. You are not injected with radioactive element labeled glucose or any other material, but you are given a dye coloring substance through injection, which gives contrast. Contrast agents may be injected intravenously or into a joint to enhance the image and facilitate. For example, if there is a problem here in cerebrospinal fluid, you may be given a fluid that runs through the spinal cord into cerebrospinal fluid, so that the color it makes your brain and surrounding to have unique color, so that if there is a problem, that problematic area can be easily imaged, or its image can be easily taken. Therefore, the substance that is injected into your blood is not emitting radiation. It is used to produce contrast for better image. Contrast for better image. You see, white image is good when the background is black. If the background is white and if the image is yellow, that's not good contrast. Therefore, contrasting material is injected. The other geographical position, GPS, location. It calculates, it shows you your location, where you are. It tells you not to lose your destination while you are working in a field, especially field tour. GPS, geographical positioning system. GPS is a device used to collect biogeography means the distribution of different organisms, plants and animals in an ecosystem. Biogeographical information, biological data on landscape mapping, plants, animals, humans, whatever. So that you can, therefore it's field tour. You can assess the ecosystem, study the ecosystem. And